Well, good afternoon. I'm Jan Nerger, and it's my pleasure to serve as the Dean of the College of Natural Sciences. I'm very pleased to welcome you to the Thornton Massa lecture. This is the 18th year of our hosting this lecture, and it truly is a wonderful event that we look forward to each year. Before introducing our distinguished speaker, I want to tell you a bit about the lecture series for those of you who don't know the story, and then a little bit about the benefactors themselves. So the benefactors of the Thornton Massa lecture series are the family of Bruce and Mildred Thornton and Dr. Emil Massa. And these families both shared interests in biodiversity, germplasm preservation, plant genetics, and related topics, which led them each to endow an annual lecture. One was through the College of Natural Sciences, and one through the College of Agricultural Sciences. And so early on, it was agreed by all those involved that we should merge these two endowments and jointly hold an annual public event with a very distinguished speaker, and hence the name Thornton Massa. So Bruce and Mildred Thornton shared a lifelong interest in the commitment to the study, identification, and preservation of seeds. Mildred attended CSU at the time, though it was Colorado Agricultural College, and she received her bachelor's degree in 1926 and a master's degree in botany in 1928. And she was the first woman to earn both a bachelor's and a master's degree in CSU history at that time, the first woman to do so. It was during her graduate studies that she met fellow botanist Bruce Thornton. And Bruce Thornton earned his degree in animal husbandry from CSU in 1918. And then he returned to CSU and earned a master's degree in botany and plant pathology. He served as a full-time faculty member from 1927 to 1962, and during his time in Fort Collins, he was also the director of the Colorado Seed Laboratory from 1941 to 1961, so for that 20-year span. Now, the interesting thing, that I think is interesting, a state policy during that period prohibited two family members from working at the same office. So, I mean, that's not the case today, just to let you know. And so that actually kept Mildred off the payroll for years. So she continued to work on a part-time basis, mainly working without pay all of these years. And when Bruce retired in 1961, Mildred then took over the directorship of the State Seed Laboratory, and so she had worked there intermittently for more than 20 years. Emil Massa was an orthopedic surgeon who worked for more than 30 years at St. Joseph's Hospital down in Denver. He had a really long-standing interest in plants, plant breeding, and biodiversity. He was a native of Cleveland, Ohio. He received his undergraduate degree from Denison University in Granville, Ohio. He earned his medical degree in 1953 from Northwestern in Evanston. And he entered private practice as an orthopedic surgeon in Denver in 1960 until he retired in 1991. He had an abiding sense of appreciation and wonder for the phenomena of the natural world. His interest in plant genetics was the basis for his support of many plant-oriented institutions, including the Denver Botanic Garden, and inspiration to begin this lecture series. His goal of the series was, and our goal, I think, remains, to annually engage the public in a thought-provoking conversation to honor, preserve, and investigate nature's rich vocabulary of organisms. So this afternoon, like nearly every year, we are able to have the daughters of both Bruce and Mildred Thornton and Emil Massa here to join us and to help celebrate this 18th anniversary of the lecture. Ms. Bobby Fry, over here, the light, there she is. She's right over here. She is the daughter of Bruce and Mildred Thornton. And then Heidi Massa over here, the daughter of Emil. And coming from California, coming from Chicago, and it's just such a gift to have them with us today. So I want to thank both families for their vision and their generosity in endowing the lecture series, and I think we're all pleased with what has come. So today's speaker is Harry Klee. He is, now, I was just talking to Harry, but I'll get back to that. So, as Dean of the College of Natural Sciences, one of the messages I try to convey to people about science is that it's not just something that you do and it stays in the lab, but it can create new biotechnologies, it can solve humanitarian problems, and it can even help feed us better. And I think Dr. Cleve is an accomplished scientist who has done all of these things, and on a global scale. He received his PhD in biochemistry from the University of Massachusetts and conducted postdoctoral research at the University of Washington. And he was employed by Monsanto Company for about a decade, 1984 to 1995, where he developed technologies for plant transformation and transgene expression, and he was a member of the team that developed the Roundup-resistant crops. He's worked on ethylene for the last 3 decades with an emphasis on its role in tomato fruit development. He's been a professor of horticulture at the University of Florida since 1995, where he established a program to understand the biochemistry and genetics underlying the flavor of fruit crops. His laboratory has identified many of the genes encoding important flavor synthesis activities, and that work has transitioned into a large-scale genomics approaches for improvements of tomato flavor, initially focusing on varieties of the home garden market, but expanding into the commercial germplasm as well. And what I did last night is I was looking up things about him just to see if I could learn more. And I read a science article that he published back in January of this year, and I want to really recommend that article to you. First of all of you know Science is a very prestigious journal. Second, it means that it's a short paper, and that's good for me. And so it's not going to be this lengthy thing. It's a short paper. But then afterwards, the New York Times picked it up, and they did this beautiful, I thought, a really nice article about him and how he's putting the taste back in tomatoes. So please join me in welcoming this year's Thornton Massa lecture speaker, Dr. Harry Klee.
Okay, let's see, somewhere there's a microphone. Maybe? Yes. Okay. Okay, well, I wasn't really nervous till I saw all the people who had given this talk before me. Now I'm very nervous. I'm going to start off, well, first of all, I'm going to tell you the story about what we've done with the flavor of tomatoes, something that I think most of you, if not all of you, are very interested in. The reason that I like this story, and I think the reason people like the New York Times picked it up, was because everyone cares about tomato flavor. Everybody thinks they know what a great tomato tastes like, and everybody hates commercial tomatoes. So this was a project that I took on probably 15 years ago, and we knew that it would be very difficult. Companies were not going to tackle this kind of thing because it's really a long-term investment, and the fun thing for me was that it's a really interdisciplinary project. So I get to work with a psychologist, a food scientist. In my lab, depending on what day it is, we call ourselves biochemists, geneticists, molecular biologists. It really took a lot of different expertises to come together to really make progress on that. So I hope that you'll get a feeling for what it's like to do interdisciplinary science. I'm going to start off with something maybe just because of the audience and because this is a public talk, and I want to make a point up front, and that is this stuff's really critical. When they asked me to do this, I said, well, of course. Anytime I can talk to the public and tell them how we do our science, it's a really good thing to be able to convey to people that we're trying to help the world and do it in a very systematic way. So after that science paper was published in January, the National Science Foundation, which funds our research and has for a couple of decades, put out a press release. Of course, they were very happy that we had done this. Unfortunately, Senator Rand Paul disagreed that this was an important finding, and he put it in his, what he calls his waste report. And he basically said that the government shouldn't be funding flavor research. Well, obviously, I think he's wrong. But I think, you know, that this is the spirit that I want to start off with and say that science is threatened today. We don't have enough really good people going into science careers these days, especially plant biology. And I think that there's this anti-science environment that really disturbs me. And for those of you on the left who are gloating and saying, well, you know, that's just those nutcase right-wingers, let me tell you, well, first of all, Rand Paul's not a nutcase right-winger, but for every right-wing person who's an anti-science, there's a food babe out there on the left. And so this is a general phenomenon that bothers me a lot, that science, the people are suspicious of science. So one of the things I want to do is hopefully disarm you and tell you the story of what we're really trying to do in a way that you'll understand, and hopefully Rand Paul would understand if he bothered to listen. So why do we work on flavor? First of all, I'm kind of a foodie, so I really do like food. I really like taste and flavor. So it was a really fun thing for me. I was hired at the University of Florida to take an endowed chair that was funded by a tomato growing family. So I was told that it would be in your best interest to work on tomatoes, which I was thrilled to do. And so what was the number one problem that we faced? It was flavor. And I'll show you on the next slide something that really catches that. But, you know, flavor is just not a curiosity. We're not just stamp collecting here. I really think that this is intimately tied to nutrition. And this is the argument we make. And that is very simple. People say, oh, well, I can get a good tomato. I can go to Whole Foods and pay $6 a pound. Well, there are a whole lot of people out there that I want to eat much more healthy diets who can't afford $6 a pound or won't spend $6 a pound. What we want to do is to increase the quality of the foods that you eat with one very simple idea, and that is if we make healthy foods taste better, people will eat better diets. And so we talk a lot about obesity, and you know, there's a reason people eat at McDonald's. They think it tastes good. And so if we can make healthy foods taste better, and I think we'll all agree that the tomato is the poster child for where we should focus our improvements, then I think we can have a real impact on society. So I want to show you this. This is an article that appeared in 1977 in the New Yorker, and it illustrates the problems with tomatoes. And if you look up here, you'll see insipid, blah, tough, like eating cardboard. That was 1977, and this is 2017, and people are still saying exactly the same thing. So this is a real problem that we need to fix. And yes, you can go and, you know, in season, you can go to your farmer's market and you can get a great tasting tomato, but most of the stuff, if you go to the supermarket, it's not very good, and we can do a lot better. So why is that? So first of all, we need to understand how the system works, and that is most farmers, not all, but almost all of them, are paid for yield. They don't care about flavor because they're not paid for flavor. The farmer is a businessman who has to, or woman, who has to make ends meet. And what that means is that they need yield. And so yield is what they're paid for, yield is what they care for. The breeder says, I want to give you what you want, I want to give you higher and higher yield. And so the breeder is thinking about the grower, they're not thinking about you. And so that needs to change. There are other problems. Wholesalers and retailers will almost always refrigerate the tomatoes when they get them. That's a very bad thing. Don't refrigerate your tomatoes. But the supermarkets know that, but they refrigerate them anyway. Why? Because they've got a whole bunch of boxes of tomatoes and they know they're not going to sell them for 3 or 4 days or more, and so they stick them in a refrigerator, and that ruins the flavor. So there's a problem there. But you in this audience are not without blame, because we know that if you wanted to pay that $6 a pound, you could get a pretty good tomato. Most people don't want to do that. They want to pay $1 or $2 a pound, and they want flavor. Well, you're not going to get that in January, because in this part of the country, that tomato is going to be picked in Mexico and shipped in a truck, and it's got to survive that journey, and it's got to survive in the store until you buy it, and it's not going to be very good. I personally am not a big fan of eating seasonal. You know, that if you live in the north, that can lead to a very boring diet over the winter. So this is the challenge. How are we going to understand flavor, and how are we going to fix it within the context of the existing commercial delivery system? But the bottom line here that I've gotten read is flavor is a very difficult phenotype. Flavor is really complex, as you'll see. Breeders don't know how to deal with it. And our objective is to reduce a very complex trait down to a series of molecular markers that a breeder can use in their breeding program and not have to worry about the quality of the flavor. They just worry about, are your markers there, and then you will have good flavor. So let me tell you a little bit about the program that we've started. We've really started to work with the breeders who are in my department and my university to take a different approach, to start with the consumer. And in order to do that, we need to understand what the consumer likes. And this is where we work with a psychologist who's an expert in basically getting into your brain and figuring out what you like without you even knowing that he's been there, or she in this case, she has been there. And so the term that my colleague Linda Bartoshuk uses is she calls herself a psychophysicist. Basically what that means is reducing chemical and physical inputs into emotions. So here's what we do in a nutshell. We start off with the consumer. So the very first thing that we've done, and I'll walk you through this, is we take consumer panels and we say, what do you like? And then what we do is we analyze the material that you have been given and say, what's in what you like? And then we say, okay, these are the chemicals that make you like this tomato. How does the plant make them? And what is the underlying genetics that controls how that plant makes those chemicals? We reduce it down to the level of individual genes, and then we go and turn those into molecular markers that a breeder can use. We say, this is an important gene, that's an important gene. You need to have the right combination of those genes in order to get good flavor. And then what we do is when we develop varieties, we go back to the consumer and say, how are we doing? And if they don't like it, then it's not a product. So we've initiated this in a few crops. We're furthest along in tomato. Actually, strawberry has used these techniques, and they've released a variety that has really superior flavor. My colleagues are doing it in blueberries and peaches. It's generally applicable. Okay, so let me tell you, let's start off with what is flavor. When I ask someone what's flavor, the first thing they usually think about is their mouth. Well, it turns out that the mouth is probably not the most important part of flavor. In fact, when we talk about flavor, don't mix up flavor and taste. Taste is what happens in your mouth. Flavor is what happens in your brain. So what you do is you take a series of sensory cues, and we'll talk about the tomato, and you input taste, olfaction, smell, the feel of the product in your mouth, and sometimes even what it looks like. What it looks like actually influences what you think it tastes like. That's why the flavor is so complex. That's why we work with a psychologist. So it turns out that probably the most important part of flavor is smell. And the real simple experiment for you to do when you go home tonight and have dinner, put something in your mouth and chew on it, but hold your nose closed when you're chewing. And think about what you're experiencing in your mouth. And what you're going to experience basically is what you get out of taste. And that is sweet, sour, salty, bitter. As you're ready to swallow, what you do is let your nose go. And this burst of volatile chemicals goes up, as it's shown here. It actually, as you swallow, it forces air up into your nasal epithelium, where olfactory receptors recognize those compounds, and all of that input is integrated into your brain. So there's a spot up here that lights up and says, oh, I'm eating a tomato. That's the result of all of those inputs. So if you think about this, there are only 5 classes of taste receptors. Think about all the foods that you've eaten in the last few days. You think there are more than 5 flavors that you've eaten in the last few days? Almost all of flavor comes from smell. There are 400 different olfactory receptors that recognize volatile chemicals. That's where the diversity of flavor comes from. We focus on the volatiles because we think that's where the real action is. The breeders honestly have paid attention to sugars and acids for a long time, which are the taste components, and because the assays are easy, but the volatiles they pretty much have paid no attention to. The other interesting thing about the volatiles is they're really biologically active. We can detect some of these volatiles at picomolar to nanomolar concentrations, minuscule quantities, 3, 4, 5 orders of magnitude lower than we can detect sugars and acids. So you get a lot of impact from those compounds. That's why we focus on them. So let's focus specifically on tomato. Basically, you need taste. I think, I'm not going to argue that sugar is not important. Actually, sugar is critically important. It's one of the things that we're born to like. And most people, well, all people are born hardwired to like sugar. You need a certain level of sweet in order for that tomato to taste good to you. Generally, you also need acids. And much like Chinese cuisine, you've got the sweet and the sour, and they balance. There's a reason that all these different cuisines use sweet and sour. That's the foundation, and you have to have that. But it's the smell that really gives you that unique tomato flavor. And again, if you want to test this, just do the nose trick. Go back and chew something, and then let your nose go as you swallow, and you will get that burst of tomato flavor. If you've got a good tomato, that is. So I want to show you something that we discovered a number of years ago that I think is really cool. Well, first of all, 400 volatiles. That's a hell of a lot of chemicals. We need to get the ones that are really important. We can't work on 400, and the fact is we don't need to work on 400. Many of those chemicals do not impact whether you like the tomato or not. So we need to find out which ones do. But I want to show you something that we discovered a few years ago, and that is, this is a list of the volatile chemicals, most of the volatile chemicals that ultimately impact flavor. Not necessarily liking, but flavor. And what I want to show you, if you look over on the right side, you'll see that word essential. The precursors for these chemicals, we've spent a lot of time understanding the metabolism, the biochemistry of how the plant makes those compounds. And what you're going to see is that a lot of them are derived from amino acids, but only the essential amino acids. Fatty acids, only essential fatty acids. We think that people actually like what's good for them. That is, your brain is trained to recognize chemicals that are metabolically linked to things that are essential for your diet. So how do we understand what of those 400 chemicals really are important? The way we do it is to exploit the diversity of the species. And so what we've done is over the years, we've got quite a collection of tomato varieties. Everything from wild accessions from Central America to heirloom varieties to modern varieties. We've got heirlooms that we can trace back to the 1870s. We've got this nice collection, and what I'm showing you here is just the variation within those varieties, and you'll see there's huge variation in color, shape, size. Well, there's also a tremendous variation in flavor, and that's what we exploit. There's this idea that all heirloom tomatoes must taste great because they're called heirloom. First of all, I should tell you there's no legal definition for the word heirloom. Heirloom basically means old, and somebody decided a long time ago it wasn't worth growing anymore. So there are a lot of heirloom varieties out there. Some of them are outstanding, and some of them not so much. So I'm just showing you this. This is just a range to tell you that we can get an immense difference in those tomatoes for how they taste. That's what we need. There's also huge variations in the volatile content. Don't worry about the numbers, just basically hundreds to thousands of fold differences in those volatile chemicals within the tomatoes. So here's the good news. Most of the modern cultivars really don't have very much flavor. I mean, the industry will tell you, what do you mean? Our tomatoes are great. We put them through our consumer panels. They don't taste great. We've got numbers and that we can show them that these are not good tomatoes. In general, it's because they're deficient in a lot of those chemicals. So what that means is that with traditional breeding, we can go back to those heirloom varieties of 75 to 100 years old, and we can recover the flavor just through genetic crosses. It's a lot of work, and we have to figure out how to do that, but it is possible. So because this is a public lecture, I realize there are a lot of scientists who will say, oh yeah, I know that. I want to define, I tend to slip into jargon and say that we're replacing genes. We're not replacing genes, we're replacing alleles. So alleles, for those of you who aren't up on it, are just versions of genes. They're variants. So just the same way that we all have the genes that make eyes. We have genes, and different people in this audience will have different combinations of those genes, alleles of those genes that give you different color. Flavor is exactly the same. We all have the same set of genes in all of these tomatoes, but we have good alleles and bad alleles. We need to identify the bad alleles and replace them with the good alleles. So if I slip and say I'm replacing genes, we're not using a GMO approach. We're using breeding, replacing the bad versions with the good versions. So here's what we're trying to do. We want to be able to, again, any of you who've ever grown tomatoes in your yard, and I expect quite a few of you have, know that environment tremendously influences tomatoes. The soil you grow it in, the age of the plant, how much sun, whether it's rained the day before you harvest them, all of those things. That's why breeders neglect flavor. They just throw their hands up and say it's too complicated. Well, so we need to reduce it to molecular markers that define the good alleles. So basically what we've done in the process is outlined here. We want to define the most important genes. We want to identify how those genes work. We want to basically figure out which are the most important genes. A tomato has somewhere between 25 and 30,000 genes in it. We have to figure out which are the important ones that control the flavor chemistry, identify the most desirable alleles, and then get them back. So we want to hand the breeder a set of markers and say, these are the markers you want, these will make your tomatoes taste better. So how do we do that? So we typically identify about 68 different chemicals in a tomato. So we're going out, we're harvesting our tomatoes in the field, we're bringing them back to the lab, we're pulling out a subset and grinding them up and assaying sugars, acids, and all of those volatile chemicals. And then we're taking a bunch of them to a consumer panel in our food science department. That's Charlie Sims on the left, and that's Linda Bartoshuk on the right. And we let them eat them. And we let them score them and tell us how much you like it on a measurable, repeatable scale. And then we basically are simply doing correlations. We're saying what's in the good ones, what's in the bad ones, how much is in each of those. And what we can do is we can statistically pull out a set of correlations of what's in the good ones and how much do we want in order to get the highest possible score. So we've done that, and we have basically a recipe for a great tomato. Now, I should tell you that this is not trivial. This has been going on for about 5 years. It's not easy to grow enough tomatoes for a 100-person consumer panel and get them back. Typically, a consumer panel can only eat 5 or 6 varieties in a day. So the logistics of this experiment are really tremendous. And this is where Linda Bartoshuk has really come in. Linda's really probably the world's expert on taste and smell. If any of you have ever heard of the term super tasters, Linda is the person who coined that term a number of years ago and defined what a super taster is. They're people that have very high concentrations of taste receptors in their mouth. And you may have done the test, some of you, with the bitter compound that you touch to your tongue and tells you whether you're a super taster or not. Well, Linda invented that. So we know now which of all of those chemicals are really important. Now, the question I always get asked is, oh, you're averaging over 100 people. Aren't people different? And the answer is absolutely yes. So there is not a single ideal tomato. We can differentiate. So we've done a lot of, we take demographic data on our consumers. We can measure, show that there are statistical differences in the best tomato between, for example, based on sex, ethnicity, age, and whether you're a super taster or not. So different people like different tomatoes. But we believe that the average of all of the population is going to give you a tomato that everyone in this room would agree is a really, really good tomato. It may not be your absolute favorite, but it would be a really good tomato. I spend some time in Asia during the year, and I will tell you that the Asians like different tomatoes. If you go to Japan, you'll see a pink tomato, which most Americans would look at and say, that's not ripe. And the fact is, Americans don't like that tomato nearly as much, and Japanese don't like our tomatoes as much. So we know that there are cultural differences, and how you were raised and how you use tomatoes in your cuisine determine what you like. We know there are huge differences between Hispanics and Northern European descent people. They like different tomatoes. Why? Because they're using them in different ways in their cuisine. But I still come back and say the average of the best of all of our American population will give the American people an outstanding tomato. So just to summarize, we've identified about 29 volatiles. That still sounds like a lot, but we'll get to that. That are either positively or negatively correlated with liking. The interesting thing is, some of the most abundant volatiles that are in a tomato. For example, if you ever buy those, I shouldn't tell this secret, but if you ever buy the tomatoes on the vine and you pick them up and you smell them, the smell that you're getting are 6-carbon volatiles, primarily things like hexanol, hexanal. That has absolutely no influence on the taste of the tomato. you're being fooled. All you're tasting is the trichomes, the hairs on the stem. When you pick it up, you're smelling that. Has nothing to do with flavor, but it makes you feel good. That chemical that you're smelling with those, it turns out not to be at all correlated with whether people like the tomatoes or not. So that's why you have to go in and empirically decide what are the most important chemicals for flavor. Some of the rare volatiles that you can barely detect on a gas chromatograph are some of the most important chemicals for flavor. The other interesting thing, I'm not going to go into any detail on this, but this was a really interesting finding, is that sugar is not the only thing that makes you decide whether something is sweet or not. Volatile chemicals actually amplify the sweet that's measured by your tongue in your brain. So think about, why do you always add vanilla extract when you cook like brownies or a cake or something like that? The volatiles actually make the thing taste sweeter than it is. And we've shown this pretty well. Well, that's really exciting because it actually opens up some interesting avenues that go way beyond tomato. We could potentially take fruit juices, take out a lot of the sugar, replace it with volatiles, which have virtually no calories in comparison, and you wouldn't know the difference in terms of sweetness. That has some really interesting implications for the food industry. Okay, so how do we translate that? Now we know what the recipe is, we know what the target is for making that tomato taste better. How do we get there? So we've used biochemistry and we've used genetics. And I'll just show you a tiny bit on the biochemistry and then focus on mostly the rest on genetics. So this is a list of the volatiles. Well, you say, oh my God, what does that mean? It means there's a lot of chemicals in there, and we've got to fix a significant proportion of those in order to have an impact. Well, this is where the biochemistry comes in. It turns out it's not as complicated as it looks. We spend a lot of time figuring out how those chemicals are made, and what we find is that we can group them together. So inside each of those circles are chemicals that are part of the same metabolic pathways. If we're smart, and if we focus on the right steps in that pathway, we can potentially influence whole sets of those genes with one specific gene. So what that means is that I can get almost all of my volatiles with 5 genes, if I'm very good at figuring out which ones I want. Well, if I tell a breeder that they're going to have to work on 29 genes, they're going to laugh at me and say, forget it. If I tell them 5, that becomes a much more manageable task. So we can get quite an impact on flavor with a few genes if we're careful and select the right ones. So this is the way we do it. We do what's called genome-wide association studies. And I will tell you, again, sort of another aside to the science. I actually have a visiting professorship in China. I go there twice a year. The reason I go there is because the Chinese have tons of money. They spend an absolute fortune on basic plant science. They are spending 5 times as much money on their plant science infrastructure as we are. So I said, you know, I could potentially use that. And if we're smart, we could have them pay for our research. So we set up a collaboration with a group in China that's really good at doing genome sequencing. And we said, you know, we've got 400 different varieties of tomatoes that we have done complete chemical analysis on for all of the important chemicals. And if we only knew something about the genetics that underlies that chemistry, we could get a really nice paper. And the Chinese say, ooh, good paper, that's a good idea, let's do that. So they sequenced 400 complete tomato genomes for us. And this was really a major breakthrough. This changed everything for us. We couldn't get that work funded in the United States today, but we did get it funded through a collaboration with China, and it didn't cost me a penny except a collaboration. So what do? These association studies, basically, what we do is we take the entire genome sequence and we quantify all of the chemicals and we look for associations. So what that basically means is you go through nucleotide by nucleotide on every single chromosome, and everywhere there's a difference, you say, does that difference impact any of my chemicals? And you march through the genome and you identify regions that do impact the chemicals that you care about. And you get a printout that looks a little bit like this. And what we're looking at is there are 12 chromosomes in tomato, so we've just alternated orange, green for each of the different chromosomes. And that red line that goes across means that if it goes above that line, it is highly significantly correlated. So what that tells us is that on chromosome 6, there's this locus that has a huge impact on citric acid, one of the acids that's really important for tomato flavor. On the bottom is another one. At the bottom of chromosome 9, there's a locus that affects this volatile chemical, guaiacol. So that tells us that nucleotide change, or that small group of nucleotide changes, each dot is a nucleotide change on the chromosome, that correlates with that chemical. So that allows us to map loci on the genome that affect all of the chemicals that we care about. So we've done that. We've done it for all of them. So that information is really useful. It allows us to do a couple of things. The first thing it allows us to do, of course, is get to the genes that we really care about that affect our chemicals. The other thing it does is having these complete genome sequences, we can go back and kind of do forensics. We can say, what went wrong with the modern tomato? We've got all these genome sequences. We can say, well, what have the breeders done in the last 50 to 75 years to really screw up flavor? And we've got the evidence right there on the computer screen. So we've done that. So I want to tell you a couple stories, just 2 slides that are 2 stories that actually are not, they're not related to volatiles necessarily, but they illustrate what breeders have done. The first is that we have done, we and a lot of other people know this, it's been known for years. The bigger the fruit, the less the sugar. If you want to have a really sweet tomato, you're going to go to the store and you're going to buy the grape or the cherry tomatoes. Why? Because they have more sugar than those big objects. Well, the problem is that the growers don't like small tomatoes. Why? Because they're picked by hand. And if you think about that, the grower is paid for how many pounds of round red objects they put in a box. It takes the exact same amount of time and labor to pick a big tomato as a small tomato. And so the growers say, we want the biggest tomatoes. Our consumer studies, I think most of you will probably agree with this, our consumer studies say consumers don't really want those giant tomatoes. They prefer the medium size. The giant ones are almost too big, but that's what they get because that's what the growers want because of the economic system that's been set up. The bigger the tomato, the less the sugar. So right off, you're taking a huge hit in sugar by going for bigger and bigger tomatoes. The second one is actually a really interesting gene, and this is not our work, but I think it really illustrates one of the big problems. So this is actually a rather extreme example, but this is what a typical heirloom tomato looks like as it ripens. It ripens pretty well on the bottom, and it basically ripens from the bottom to the top. And what you get is what's called a green shoulder. And most of the heirloom varieties out there have this phenotype. Well, the breeders back 100 years ago discovered a mutation. And that mutation is called uniform. And what uniform does is it removes that green shoulder and makes the fruit ripen more uniformly. Well, how does it do that? It turns out it's a gene, a transcription factor, that controls the number of chloroplasts in the fruit. And so what you're doing is you are reducing by about 20% the chloroplasts in that fruit. That's why there's less contrast, because it's less green. So it ripens up and looks very nice. And the growers said, oh, this looks better. This is what the consumers will want. Well, it turns out any of you who've had botany or even biology 101 will know, what does a chloroplast do? It takes light and light and CO2 and turns it into sugar. So you have a mutant that has 20% less factories making sugar, and you get less sugar in that tomato. So right off, and the sugars in turn are the foundation for a lot of the other chemicals that give you flavor. So right off, you are knocking down the flavor in that tomato because someone decided that this looked nicer. So that's part of why some of the heirlooms taste much better. So this is, again, it's a case of a disconnect between the consumer and the breeder. Okay, so let's talk specifically about the chemistry of the modern varieties. Having the complete genome sequences of all these allow us to do these phylogenetic trees. We can separate out the modern varieties from the older ones, and we could say what's different. So it turns out when we do that, this graph is actually not as complicated as it looks. 0 means that there is no difference between all of the modern varieties and all of the older varieties in all of these different chemicals. Well, what you'll see is about half of the important flavor chemicals are significantly lower in the modern varieties. Again, that's what I told you at the beginning. This is just the data that shows it. Way lower. So think about that. Half of the chemicals are lower. Think about if I took a coin, so I've got 2 alleles of a gene. One of them is a good allele, one of them is a bad allele. I flip a coin 30 times, because there are 30 molecules, let's say 30 genes affecting flavor, and about half the time I'm going to land in the wrong way. Well, that's exactly what's happened, is that we have lost, basically through a lack of positive selection, we end up segregating away the better alleles for a lot of the flavor genes. Now, this has occurred over probably post-World War II is probably when they started really intensively breeding tomatoes. So we're talking about the last 75 years or so. It's not a sudden process. It's a gradual process. So the way that I'm going to describe it to you is by showing you a picture of a symphony orchestra. And I'm going to tell you that there are between maybe 80 and 90 different instruments up there. And imagine you're listening to Beethoven's Fifth Symphony, and suddenly one of those second violins gets up and leaves. And maybe a flute leaves, and maybe one instrument here, one instrument there, over an extended period of time. I think most of you would not notice the difference if one instrument left. Well, think about the flavor the same way. Over the last 75 years, the modern varieties are extremely narrow genetically. The breeders have exchanged material or stolen material. Most of the modern varieties there's almost no difference in them. People have lost the best alleles one at a time, and then 75 years later we wake up and say, oh my God, what happened? Flavor's gone. Well, it's because the tomato flavor is the combination of all of those chemicals, and when you start depleting them one at a time, and you're not really removing them, you're just pushing them down, it would be almost impossible for a breeder or a grower to even go out and say, oh my God, I've only got half as much guaiacol. It's not going to happen. So that's what's happened. You've basically, almost by random chance, you've lost the good alleles. So I'll start with the bad news. The growers are not going to accept debater tomato. So we know how to do this now. We've figured out the right genes, the right alleles, and all we have to do is get them back in. The growers are not going to accept that tomato, even if it tastes 10 times better, unless I can do that without destroying yield. So that is a big challenge for us, and the good news is we know how to do it, but it's going to take time because we have to put these genes in a few at a time and get them in and significantly impact the flavor profiles, but we have to always go back and say, is the yield equivalent? Is the shelf life equivalent? And if it isn't, they're going to say, forget it, I don't care. You could change that. We are trying to change that through consumer pull-through, and you know, things are changing. I'm more optimistic than I used to be. You know, the two things that I always throw out as examples are the emergence of craft beers and the emergence of Starbucks. And you guys, I'm sure there's someone in the audience who says, oh, Starbucks, that's not that good. Well, 30 years ago, Starbucks was a hell of a lot better. And it has become now the gold standard. And yes, you can get better, but it's a pretty good cup of coffee. And so people are starting to pay attention to flavor. A lot of people are willing to pay more money for flavor, but we still, where I started is where I want to end, and that is we want to impact the cheap tomato, the industrial produced tomato, because I don't care if you make $200,000 a year and you can afford to shop at Whole Foods. I care about the people on food stamps who can't afford to shop at Whole Foods, and I want to get the better tomato to them. And the only way we're going to do it is to put the flavor back in without compromising yield. And I think we can do it, but it's going to be, it's basically, I know exactly what needs to be done, and it's just a matter of turning the crank. So I want to just wrap up by saying that, no pun intended, there are some lower hanging fruit that we can get, and that happens to be the home garden. Now, how many of you grow tomatoes in your yards? How many of you grow Better Boy? A few. Well, the dirty little secret is Better Boy is actually a commercial variety that was developed that wasn't quite good enough. It's not a bad tomato, and when you grow it in your yard, it's not bad, but we can do better. So what we have done is we've said, we've done all of this research about which heirloom varieties taste the best. What if I can give you something that has heirloom flavor and grows as well as Better Boy? And Better Boy grows really well. It's a very versatile tomato. It's the #1 selling tomato in the home garden market in the country. So what we decided was, what if we took a modern elite inbred variety that was developed at the university that has good disease resistance, great yield, good shelf life. It's just a workhorse tomato, and we crossed it to one of the best tasting heirlooms. What would happen? And it turns out that we've done a lot of these, and they don't always work. In fact, many of them we've thrown away for one reason or another. But occasionally, we hit on something where the hybrid tastes as good as the heirloom parent, and it way outperforms the heirloom parent in terms of yield. So we've done that. We've actually got 3 separate varieties that we've released, and they all outperform Better Boy. They all taste way better than Better Boy. And so for the home gardener, we've got stuff that's really significantly better. So we've sent these tomatoes out to over 10,000 different people now. And I would say 90% of the people love them. 10% say it wasn't as good. In general, we've been very pleased with it. Well, what we've done is we've taken some of the best ones And we're trying to work backwards to, again, this pull through the system. So we've sent them to a lot of organic gardeners. We've sent them to some of the big companies to trial. And in general, when they do that, they really like them. They can sell this tomato. So if you know someone who's growing for a farmer's market, I can give them something that gives twice the yield and tastes just as good as what they're selling you today. In theory, they should be able to drop the price from $5 down to $2.50. They probably won't, but they could. So we're trying to get it out that way. We're trying to build the demand for flavor. And we're making progress. We had a big grower in the East grow 1,000 plants this summer, and they've sold all the tomatoes they could make. But they're selling them at a premium. Okay, so just to sort of wrap up, we know exactly what makes you like what it is, about that tomato. We know what the chemistry is of liking. And I should say we've also done this with blueberries. We've done it with strawberries. Strawberry is the other one that I'm sure most people would agree really is pretty lousy in the store. We know what drives people to like strawberries, blueberries. We've done peaches, we've done cantaloupes. We have a chemical fingerprint for great flavor for a lot of different fruits. We know what's been lost during breeding, and we know how to put it back in. We've started that process. Again, when you're using traditional breeding, it takes time. You can only make the plant grow so fast, and you can only get the right gene in and get rid of all the other stuff that's not supposed to be there so quickly. I know it's controversial, but I will at least tell you that we are not using GMO. If we could use GMO, if we could afford to, I could make that tomato for you today. And you would agree that it was a great tomato. We're not going to do that. It's too expensive and too controversial. So we're doing it by traditional methods, but we know how to do it, but it takes a lot of time. Probably 2 to 3 years, I think potentially in 2 years, we may have stuff that we think tastes really good. The thing about reducing calories, reducing sugars, is actually really cool. It was an unexpected finding. This is serendipity, and it really works. And we've gotten some of the food companies to express some interest in it. We're talking with them about how they could potentially make healthier products with less sugar. And we've released some heirloom hybrids, but finally, the point is, what we've done with tomato will work for other crops, and it's a very exciting opportunity for us. We really want to make stuff that you people will really want to buy. And I need to acknowledge a few people, Denise Tiemann in my lab, who's been with me for 20 years and has been at the heart and soul of all of this project. Linda is a fabulous psychologist. She's, again, the world's expert on flavor. Charlie is a food scientist. And my collaborators in other places, I particularly want to point out San Wen, who's in the Chinese Academy of Agricultural Sciences in Beijing. We couldn't have done any of this stuff, we couldn't have done the genetics without his 400 genome sequences. And finally, I want to acknowledge Liz Baldwin. Liz has worked tirelessly on tomato flavor for longer than I have and was really kind of an inspiration. We've talked a lot. We don't directly collaborate. She's a USDA scientist in Florida, but she's really been a pioneer in pushing me to work on what we do with flavor. And we couldn't have done this work, just regardless of what Rand Paul says. We couldn't have done this work without constant funding from the NSF. It just wouldn't have happened. NSF is something that has allowed us to take the real long road and not have to deliver something tomorrow to make a profit. And they've been great partners and supported us forever. And with that, I will stop.
So we have the opportunity for some questions now, and I've got the microphone here if anyone would like to start. Here. I saw this hand first. So.
Thank you so much. Very interesting. Can you go back or maybe expand a little bit on flavors? Do we like the same flavor for tomatoes for cooking, like for marinara or on pizza, versus a pico de gallo or something you're throwing on your salad? Is that the same tomato?
No.
Okay.
No, it's a good question. And so I sort of alluded to that with the Hispanic versus Northern European kind of differences. So the Hispanics like a tomato that's less sweet and more acid. Why? Because they're chopping it up when it's less ripe and putting it into salsa, whereas the Northern European types generally slice them, put it in a salad, or put it on a sandwich. We're using it for different purposes. And I'll tell you that I haven't talked at all about processing tomatoes that you'd use for like a marinara. The processing tomatoes in cans are treated totally differently. In fact, the breeders who develop those varieties are very distinct breeders. There's almost no crosstalk. The first thing they do when they harvest all of the tomatoes for making paste is they drive off most of the water to concentrate it down. Well, all the volatiles go away with the water. So, you know, and I've asked them, why don't you capture those volatiles and add them back? And they say, well, because we're going to add some basil and some oregano for the flavor. So really all they're doing is concentrating it down to sugars. So totally different products. So there are huge differences in how we use the tomatoes that impact the chemistry, yes.
So this hybrid heirloom, how about their shelf life?
So it turns out that a lot of the heirloom, well, not a lot, but some of the heirlooms, we were grinding them up. So the other thing we've done, I didn't talk about, is we have a predictive model. We can take the chemistry of a tomato and predict how much you'll like it. And some of them were outliers, and we couldn't figure out what was wrong. We'd say, well, this one should taste great, and people hate it. In almost all the cases, it was texture. A lot of those heirlooms are really soft, you know, like a brandy wine. If I gave you a brandy wine picked off of the plant today, right when it was just ripe, I think most people in this room would say, that's a great tomato. 48 hours later, you'd hate it because it goes to mush. And so actually bringing in the modern variety really brings in firmness that really helps a lot. They can't be as crisp as apples, but really the added firmness that the modern variety brings actually makes it better. So they have decent shelf life. These tomatoes have shelf life comparable, not as good as that 99-cent a pound tomato that you can sit on your shelf for 2 weeks, but they're pretty good.
You had mentioned that you've also looked at some of these early varieties, primitive varieties from Central and presumably Northern South America. Do they carry most of these desirable alleles, or are many of these alleles mutations that have taken place since domestication?
Great question. They're all there in the wild stuff. And it's just been lost through selection. And actually, those GWAS studies that I showed you, you can identify whole regions of chromosomes that were basically thrown away as people selected out, particularly for fruit weight. Fruit weight is the biggest thing. I should have said that. I don't blame the modern breeders for bigger and bigger. It actually started with the Mayas and goes way back to pre-Columbian days. people have been selecting for bigger and bigger fruit, and you can identify those regions of the chromosome that were under positive human selection. And what you see is, no, they are not mutations. They are just selecting out bad combinations of alleles.
I've been around since the end of the war, and it seems like living in different parts of the country and overseas, the two big tomatoes that seem to be very good were the Creole tomatoes in Louisiana and the beefsteak tomatoes, some of which we brought in from France. Those are big tomatoes, but they've got a lot of flavor, and I don't know what happened to those, if it was difficult to breed those or if it just didn't. Seems to me when I was a kid and as we grew up in Louisiana, Creole tomatoes last a long time. I mean, they don't go to mush. They stay pretty good.
Well, the big thing is yield. The growers are not going to touch them if the yield is not up to what the modern varieties are. And there's no doubt the yields have been going up, up, up. And I should, there are good tomatoes out there, but they generally don't have the disease resistances. They don't have the yield. Sometimes, for a long time, like the tomato industry in Florida is run by a cartel. It's legal. But no, it's totally legal, actually. They've got, I forget what the proper term is, but they're allowed to basically set prices and set size standards. And I don't know if any of you have ever heard of the ugly ripe tomato. The ugly ripe is something that was developed. It's actually a modern variety, but it's got all those kinds of indentations on it that makes it look like an heirloom. The Florida tomato industry actually sued the growers that were growing it because they said it didn't meet our specifications. So they have specific size. It has to be perfectly round. And probably the tomatoes that you're referring to, like the beefsteaks in particular, they wouldn't meet the standards. So they'd be thrown out.
Thank you. So in 2 or 3 years, when you have your great tomato, what will it be called and how will we find out about it, since it's our tax dollars at work?
To get the maximum impact, we have to deal with a company. We'll have to deal with, ideally, I should say we have not patented any of this stuff, and we have no intention of patenting any of this stuff. We are going to give this stuff out to all of the companies, and they will each name their varieties whatever they want. We finally reached an agreement with one of the companies. They're actually going to put a postdoc in my lab to help them introduce these genes back into their varieties. I don't know what they'll call them. That's up to them. Now, for our home garden varieties, if you want to donate enough, there's a naming opportunity.
Thanks, Harry. Very nice talk. I'm intrigued by the correlation you had between the volatiles and the precursors that were all essential amino acids or essential oils. So are these great tasting tomatoes more nutritious? And are the modern tomatoes not very nutritious?
That's a dangerous thing. So I'm going to tell you something, but don't take it the wrong way. I've talked about flavor. Nutrition, you could have substituted micronutrients for flavor and I could have given you the exact same talk. People aren't selecting for micronutrients. So there's some really nice papers that have been published that show that in wheat, for example, the levels of some of the important micronutrients are lower in the newest varieties versus 30, 40-year-old varieties grown side by side in replicated trials. The one micronutrient that is actually still high in the tomato is lycopene. Why? Because it's red. And that's something a breeder can look at and say, oh, yeah, it's red, I want that. In fact, we have a mutation that actually enhances the accumulation of lycopene. But in general, the answer to your question is yes, they are lower. The reason I don't like to say that in talks is because I don't want people to think, oh my God, the tomato isn't nutritious. The tomato is still a very, very good thing for you to eat. It's not the highest fruit in vitamin C, but because of our intake, it's the #1 intake source of vitamin C in the American diet. So yeah, maybe they're 10%, maybe they're even 15% lower in some of those important nutrients, but that doesn't mean you shouldn't eat it. And I just don't want to scare people into thinking they shouldn't eat them because they're not healthy. They're still healthy, they're just not as healthy as they possibly could be because we haven't screened for these things. And again, we've just, as you select for higher yield and you don't select for nutrient content, you will lose it.
Early in your talk, you mentioned that refrigeration is bad in terms of reducing flavor.
Yeah.
Is that true for other fruits as well? Or are there some fruits that lose flavor when you refrigerate them and others that don't?
It's certainly true for tomato. Tomato's a tropical fruit, and it's not ever meant to be chilled. Apples, you can chill them till the cows come home, and they won't change. Peaches are one that I do know is are sensitive to chilling. They don't recover well from the chilling. But I would say that a lot of our temperate fruits are perfectly happy to be chilled, and that's the way you get a Granny Smith apple all year round. You know, they stick them in a 4-degree room with controlled atmosphere. But certain ones, I know that the tomato is bad. I know that the peach is bad. I know some others are not bad, but I'd have to go down the list.
I have to ask the question in relationship to, I work with greenhouse growers quite a bit, and when you start working with the large commercial varieties, are you looking at also correlations with cat facing and skin thickness and shoulder cracking, and do those all come into effect as well?
So for the home garden, the simple answer is no. For the commercial, absolutely yes. So what we're doing is we are starting with a very elite variety of tomato that was developed by our tomato breeder, Jay Scott, a number of years ago, well, 10 years ago or so. And those are very important traits. Should they be? We could argue about that all day. But yeah, it's very important that they be round and that they have a small blossom scar. They're not susceptible to cracking. All of those things factor in, or again, they're not going to touch them. And that's what makes it so hard. So, I mean, I mentioned a couple of traits, but you're right. There are many more traits that we worry about.
Have you taken into consideration of splicing with maybe peppers or other fruits to expand the sweetness or flavor?
You mean, I'm not sure what you mean by splicing. Maybe you should be a little more...
Like taking the taste of alleles from maybe something minor, pepper, to enhance the flavor.
So we're not going to be able to do that without genetic engineering. We could do that. You know, there's what is the classic Simpsons? I think he took the THC pathway and put it into tomatoes in one of their episodes. No, we are not. Look, I mean, I'm very proud of the time that I spent at Monsanto and the efforts that I did to develop crops that are covering 90% of the United States. But I'm not going to go there with these crops because I can't afford to, and I don't want to fight the battle. with the anti-GMO people to get a better tomato. So you're not going to be able to get that kind of diversity that you're asking about without, I mean, the genetic barriers are too great. You're going to have to take the gene and you're going to have to clone it out and then transfer it and make a new product. And then you'd have to go through all sorts of regulatory approval. It's a great idea. We'd love to expand the flavor potential of these things, but we just can't do that.
Hi, great talk, thank you. Being that you're in Florida, fruit obviously grows better there. I'm curious about the home garden varieties that you developed and if we would be able to grow those here expecting the same quality and flavor, or do they require special greenhouse conditions?
Yeah, another excellent question. So the simple answer is yes. You can, and I'm sure that a number of people have. We have sent these tomatoes to people in all 50 states. There are a couple of areas where people don't seem to do as well in the high plains. So we've got people like in like Flagstaff, Arizona said, oh, our season is so short, they don't mature fast enough. Someone in Idaho told us the same thing. But across the Midwest, across the Northeast, and across the Pacific, they've done fabulously. And what I would say is, Florida is actually a terrible place to grow tomatoes. If you've ever been there, it's hot, it's humid, it rains a lot, and the soil is nothing but sand. In fact, we would argue that if you can grow these tomatoes in Florida, you can probably grow them almost anywhere. It's actually a pretty tough proving ground for tomatoes. There's heavy disease pressure. There's absolutely no organic matter in the soil. And they do well there. So in many ways, we see that as sort of the gauntlet that it has to do. But we've had really good reports. Again, I won't say everybody is thrilled with them, but for the most part, actually, if you go, you can look at I don't do Facebook, but my tomato has a Facebook page. So if you go to Facebook and do Garden Gem, you can actually look at the comments of people from around the country of what they thought of them. But I think in this area they would do very well.
Hi. I'm curious about the applicability of the results, or at least the methods, to the improvement of other crops. Mm-hmm. I understand that probably it's pretty applicable to sweet, acidic fruits, but do you think it's beyond that to grains, legumes, and other things, or how would you do it differently?
Well, it has worked in strawberries, and strawberries are a real challenge to a breeder because they're octoploids. And it's very easy for us to identify, well, it's not very easy, but we can identify in a fairly straightforward way which chemicals are important. I would say that for some of these crops, what's going to be limiting is how much genome sequence do you have available to you. That's going to be a challenge. You know, there aren't very many, I mean, sequencing is getting cheaper every day, but you do need a fair investment in understanding the genomes of these crops to do it. But I think it'll work for anything that you're willing, well, two things, that you have the genome sequence and that you have enough variability in what you're testing. If everything is very narrow in what you test, you're not going to be able to pick out what's really important. So you need that diversity. We're lucky with tomato. We can do that with strawberry. We can do that with blueberry. There's a lot of variation. We've done it with peach, where there's quite a lot of variation in flavor. But peach, we're not going to be able to, today, we couldn't start a breeding program that would improve peaches, for example. We can tell you which varieties people will like the most, for sure. And we're doing that actually with some of the retailers. But the genetic approaches, you know, the crops that it's going to be most amenable to are the ones with the most genetic resources.
In my garden, I can pull the weeds, and my daughter can too, but I know about the Roundup controversy, and I was wondering if you had any comment on Carrie Gillam's new book, Whitewash, the story of a weed killer, cancer, and the corruption of science. It seems to me that because of the active ingredient in Roundup,
glyphosate,
it seems to be a problem. You said that you were proud of your work there of
Yep.
making things that were resistant to,
Yep.
I presume that. But for we who are gardening, we don't have that big of a problem, but breeders and I guess the other people must. And can you talk about that a little bit?
Sure. You know, it's not anymore my direct area, but I'm familiar. I haven't read that particular book, but I know what it's based on. There was an organization that issued a report that labeled it as a probable carcinogen. Everybody else in the world disagrees with that report. And there actually are, it's based on a study that was flawed, that was pulled out of the journal in which it was published. The journal retracted the article because it wasn't properly controlled. There's actually, just in the last couple of weeks, and I can't remember what journal it was maybe in Forbes or one of the business journals, there was actually a story, an expose about the guy in the US who, I don't know if it's that guy or not, but the guy in the US who was their statistician, you should read that article. I mean, basically it suggests that the report that came out of this agency was highly flawed and probably not that, it was incredibly biased. The bottom line is, I think, and any farmer would, I think, agree with this, that glyphosate is probably one of the safest chemicals ever to have come out of industry, and it is not a carcinogen. I mean, that's the bottom line. There's one study by one group at one time that said they found more tumors in mice in a badly controlled experiment. I don't believe glyphosate is dangerous, and I think I'm not going to defend all herbicides. I can tell you right now there are herbicides that I would not use. I would not use in my yard atrazine for a number of reasons. I'd be reluctant to use 2,4-D. I have no problem at all with using glyphosate. It is, all of the studies that are valid say that it is no more toxic to humans than table salt. So I disagree with the foundations of the book. I think there's a good reason that Roundup-ready crops are grown around the world on so many acres, and that's because I think the study is flawed. Yeah.
Well, I was wondering if you had any insights on what's going on with wheat. With wheat?
Oh, yeah.
Back in my grandparents' day, wheat was the staff of life.
Yeah.
And now half the people I know won't eat it because they say they feel better when they don't. You know, maybe it's gluten, maybe there's a change in the protein, but something's going on.
Yeah, I mean, I don't know that much about wheat. I can tell you that the studies do say that it is slightly less nutritious than it was in past years, but it's, you know, it's still not bad. I guess I don't want to touch the gluten controversy. It's not my area. You know, there clearly are 2% of the public that have celiac disease that truly are sensitive to gluten, and there probably are some more people that are sensitive to gluten, but I still eat it. Yeah, I guess I don't want to go there because I'm not an expert on it.
So if they're doing, oh, one more, sorry.
You know, one thing that hasn't been talked about so much in terms of the components of the consumer experience is the texture idea. Mm-hmm. I don't know about other people here, but texture is usually the deal breaker to me.
Oh, it's hugely important.
And I'm wondering what you were finding in terms of whether the what is it that's affecting our texture, and does any of your work impact that?
So we have not looked directly at texture ourselves. Again, I mean, texture probably is as complicated as flavor is. The texture is hugely important when it's bad. So I mean, what we found is, again, to go back to those predictive studies we've done, we could predict accurately 85% of the time how much people would like the tomatoes. The 15% that we could not predict were mealy, too soft, just didn't feel right in your mouth. We know texture is really important. I didn't go into this, but some of the volatile chemicals that are really important for flavor are actually generated in your mouth when you chew on the fruit. And so the texture of the fruit determines how much of those volatile chemicals escape from that fruit. So we know it's hugely important, but I would argue that our data in tomato, I mean, probably I can summarize it by saying it's a huge problem if it's bad. But for most tomatoes, there's a very broad acceptable range, let's put it that way. You can't have it be super crisp, but it clearly impacts, so we've done some peach studies, and we've looked at what's called melting flesh versus non-melting flesh. The breeder in our department was developing these non-melting peach varieties. You could cut them open, they smell beautifully, they have lots of sugar. You bite into it and it tastes like an apple. Well, I mean, it feels like an apple. And I don't like them. And our consumer studies say people don't like them. And so there's a case, texture has a huge impact on your liking of the product. At least for the tomato, we can say that in most of them, it's not a problem. But there are some, we throw some out because they just are too soft or too mealy. Mealy is a big one. I'm not sure everybody probably knows what mealy means. It's kind of like that gritty kind of taste that you get. And that's a killer. If it's gritty, if it's what we call mealy, it's dead. So it can be a huge problem, and it has a major impact on whether people like them or not. I'll stay here all night if you want.
We have more.
I'm not going anywhere.
I'd like to jump back to the super tasters.
Yeah.
Is it related to, I know there's most of us are playing with 23andMe and doing the Ancestry.com genetic analyses, and we're now starting to see people getting reports on their taste capacity, especially with cilantro.
Yeah.
And for instance, I came back as craving certain kinds of salty snacks and such like that. And is there differentiation with the super tasters at the genetic level, like it is with cilantro, that is like two spots on allele?
So the simple answer is I'm not sure. We do know, so if you've ever done the super taster test, has anybody here done that test? That test measures one bitter taste receptor. And it is generally correlated with the density of what we call spongiform papillae on your tongue. The more bitter it tastes, the more of those receptors you have. So we know the receptor. So do we know a mutation or a SNP that's correlated with super tasters? I'm not sure. But we kind of know the chemistry of it, but I'm not sure if we know the genetics or not. I can tell you that there is a marker for one of the volatile receptors. One of the volatiles called beta-ionone, which is derived from cleavage of beta-carotene. There is a marker for that, and you can predict 25% of the population cannot smell it at all. And yet it's one of the most important volatiles in a tomato. And that one I know there is a marker for. So there is a growing area of what people are calling functional foods that aims to match up genetics to preferences. It's a growing area, but I don't think that there are a lot of markers. I know of the 400 volatile genes, there are only a couple that actually have been correlated with their receptors, with specific chemicals. That's a good question. I'll have to look into that. I don't know. I'm going to look into that.
Okay, well, thank you all very much for coming this afternoon.